The role of evolving technology and laboratory methods. High-level descriptions of complex phenomena versus detailed understanding of mechanisms. In the mid-19th century, pioneers who began to approach the secrets of the natural world through scientific discovery were limited by the absence of available scientific knowledge about the inner workings of cells. Despite that limitation, both Charles Darwin and Gregor Mendel used observations and experiments to penetrate some of these mysteries, but their progress was restricted by the few tools that were available at the time. The tools of discovery include methods that lead to new empirical discoveries, as well as advancing the conceptual framework that depends on that knowledge base. The impact of new methods and technology from 1928 to the 21st century. The following list will briefly summarize how innovative laboratory methods have resulted in major advances in genetics and our understanding of the genome. Several of these topics will be discussed and referenced in more detail in later chapters. 1. In the first decades of the 20th century, genetic experiments in the fruitfly, Drosophila, led to major advances in Mendelian genetics. 2. In 1928, an unknown transformation factor in bacteria was implicated to be the genetic material. In 1944, experiments that used purified enzymes to destroy specific macromolecules, such as DNA or proteins, demonstrated that DNA is the genetic material. 3. In 1952, DNA was confirmed to be the genetic material by labeling the proteins and DNA of bacteriophage, viruses that prey on bacteria, with either radioactive sulfur or radioactive phosphorus to label proteins or DNA, respectively. This demonstrated that the genetic material from the viruses that entered the bacterial cells was DNA. 4. In 1953, X-ray diffraction data of DNA, combined with published information regarding the ratios of the 4 nucleotides that make up DNA, led to the discovery of the double helical structure of DNA and suggested a copying mechanism that would allow DNA sequences to be replicated during cell division or reproduction. 5. In 1961, radioactive labeling and cell fractionation techniques demonstrated that the information in DNA is transcribed into a type of RNA called messenger RNA, which directs the synthesis of specific protein chains at cellular structures called ribosomes. Each position of the messenger RNA sequence consists of one of 4 possible nucleotides, A, C, G, or U. 6. Also in 1961, cell-free preparations of RNA consisting of long synthetic sequences of U nucleotides were used to determine that triplets of UUU specify the addition of the amino acid phenylalanine to a growing protein chain. Each protein chain consists of a sequence of amino acids. 20 possible amino acids are located at each position in the amino acid sequence. 7. In messenger RNA, each sequential group of 3 nucleotides, called a codon, specifies the identity of each amino acid in the growing protein chain. By 1966, similar techniques were used to decipher all 64 possible triplets that specify one of 20 amino acids, or the end of the amino acid sequence. This correspondence between codons and amino acids is called the genetic code. In 1972, it was thought that this non-coding DNA was evolutionary junk that had no biological functions, but represented raw material for evolution. Later, techniques that allowed isolation and characterization of individual DNA sequences demonstrated that so-called junk DNA actually has numerous important functions. 9. In 1970, it became clear that large numbers of genes arise by duplication and divergence of existing genes, an important evolutionary process referred to as gene duplication. 10. In 1970, basic research in viral genomes led to the discovery of retroviruses. Isolation and cloning of the reverse transcriptase enzyme made it possible to create DNA copies, called complementary or cDNA, from RNA templates. This later made it possible to sequence and characterize RNA as well as DNA, and to distinguish the protein coding portion of the genome. 11. Basic research in bacterial and bacteriophage genetics led to the recombinant DNA revolution, which began in 1973. isolation of restriction enzymes from other bacterial strains, which cut DNA sequences at specific short recognition sequences, such as GAATTC, made it possible to insert DNA from any organism into bacterial or bacteriophage cells, replicate the foreign DNA, and then isolate molecular clones containing large numbers of copies of a particular sequence. Once segments of DNA could be cloned, their specific sequences and molecular interactions could be characterized in great detail. 12. In 1975, comparisons of DNA and protein sequence information from humans with that of chimpanzees demonstrated that their protein coding sequences are so similar that it is likely that the differences are due to regulatory mutations in DNA, that is, mutations that control the expression of protein sequences. 13. In 1977, powerful DNA sequencing techniques were developed. 14. The development of the Internet Protocol in 1982, along with databases for storing DNA, RNA, and protein sequence data, led to the storage of massive amounts of publicly available sequence information from a variety of organisms. 15. The development of powerful new software methods for sequence alignment and comparison made it possible to determine the precise evolutionary lineages of related organisms. 16. In 1987, the invention of the polymerase chain reaction, PCR, made it possible to rapidly amplify specific DNA sequences with far less effort and resources than molecular cloning. 17. By 1987, enough sequence information had accumulated to identify the mechanism responsible for expansion of genomic DNA with repetitive sequences, known as slipped-strand mispairing or replication slippage. 18. In the late 1980s and early 1990s, combining systematic genetic screening in Drosophila for mutations that affect development with new molecular techniques that allowed visualization of gene expression in Drosophila embryos led to a new understanding of the genes responsible for the formation of the entire body plan of the flies during development. 19. In the 1990s, comparisons of related DNA sequences demonstrated that very similar mechanisms control the development of a broad range of animals, including both invertebrates such as Drosophila and vertebrates such as frogs, mice, and human beings. 20. Beginning primarily in the 1990s, a variety of powerful new techniques in molecular, cellular, and developmental biology, major conceptual advances led to the relatively new field of evolutionary developmental biology, Evo-Devo. 21. By 2001, improvements in rapid sequencing technology made it possible to sequence the entire human genome. Since that time, it has become more and more practical to sequence the entire genome of a variety of organisms and use powerful software techniques, bioinformatics, to compare them. 22. In the 21st century, a variety of molecular, cellular, and developmental techniques are routinely combined in biological laboratories to probe the mechanisms of genomic evolution, as well as the development of structure and function from genetic information.